All right, let's dive into a real molecular detective story. This is a groundbreaking investigation that connects a super common microbe living inside all of us to the very origins of cancer. For years, scientists were totally stumped by a phantom culprit, a cryptic clue, and a crime scene hidden deep within our own DNA. But now, thanks to some absolutely brilliant molecular forensics, the case is finally being cracked. Think about this for a second. What if one of the trillions of bacteria chilling in your gut wasn't just a harmless resident, but a highly sophisticated criminal? One that's capable of breaking into your cells and vandalizing your DNA. It sounds like something out of science fiction, but for a long time, this was a very real and a very urgent scientific mystery. So, every good investigation starts with a clue, right? Well, this one began with a really crucial piece of evidence. In the DNA of some colorectal cancer tumors, scientists found this distinct pattern of damage. They called it a mutational signature. And this signature was just like a criminal's fingerprint left at the scene of the crime. It could uniquely identify the bad guy, even if nobody knew who they were yet. And that fingerprint? It led investigators straight to a prime suspect, a very specific strain of E. coli bacteria. It's known as PKS-positive E. coli, and these little microbes, well, they're found way more often in colorectal cancer patients. And they were already known to produce some kind of mysterious DNA-damaging toxin. The question was, could they prove it? Could they definitively link this suspect to the fingerprints? So the investigation started to zero in on the weapon itself, this elusive toxin that the bacteria was making, a molecule with the name Colibactin. Colibactin is what's known as a genotoxin, which basically means it poisons your genes. And its method is brutally effective. It creates something called an interstrand crosslink. The best way to picture this is to imagine it literally handcuffing the two strands of your DNA helix together. And once that happens, it's impossible for your cells to read or replicate their own genetic code. It's a huge problem. But here was the real problem, the reason this case just went cold for so long. Colibactin is basically a molecular ghost. I mean, it is so chemically unstable that it breaks down almost the instant it's made. You couldn't isolate it, you couldn't put it under a microscope, you couldn't study it. So how in the world do you catch a culprit you can't even see? Well, how do you catch a ghost? You don't. Instead, you devise a brilliant sting operation. And that's exactly what these scientists did. They came up with a plan to catch Colibactin in the very act of damaging DNA. Here's how their little experimental heist went down. First, they took their prime suspect, the PKS E. coli, and they fed it some bait, just short, known strands of DNA. Next, they just let the bacteria do its thing, produce its Colibactin toxin, and let it attack and handcuff that DNA. Then came the really crucial step. They fished out only the handcuffed DNA and used some super advanced techniques to map the exact locations of the damage. They were essentially creating a molecular crime scene map. And that crime scene map, it revealed their first major clue. Turns out, Colibactin wasn't just attacking randomly. It had a very clear preference, a signature move. The data showed it was overwhelmingly targeting DNA sequences that were rich in the bases adenine, or A, and thymine, T. This was its MO. So the next clue they uncovered was the culprit's point of entry. You see, the DNA double helix has these two grooves that run along its length, a wide one and a narrow one. Using molecules to block these grooves, the scientists found that when they blocked that narrow channel, the minor groove, Colibactin couldn't do any damage. They had found its secret passage. Okay, so let's just recap. The investigators had the suspect's fingerprint, they knew its preferred target, and they even knew its secret point of entry. All the evidence was pointing to one conclusion. It was time for the final reveal. The first ever clear picture, a molecular mugshot of Colibactin caught red-handed. So after all of this incredible work, what did the scientists finally see? What does this molecular ghost, this unstable phantom, actually look like when it's finally caught and bound to its victim? Well, the answer began with this number. Using a super high-precision molecular scale, it's called a mass spectrometer. They measured the exact weight that Colibactin added to the DNA, and the result was 771.26 daltons. Now that's a tiny unit of atomic mass. But this number was a complete surprise. It didn't match the weight of the molecule they thought they were looking for. This single, unexpected measurement, this was the clue that blew the case wide open. And here is the big twist in our story. The very thing that made Colibactin a molecular ghost, its incredible instability, that wasn't a weakness. Turns out it was its secret weapon. A highly reactive chemical group right there in the molecule's center was the key to its entire destructive power. Yeah, understanding this unstable center was the linchpin. It was the key that allowed scientists to finally connect all the dots and crack this case for good. And to prove it, they did something really clever. They created a stable imposter Colibactin in the lab, one that was missing that unstable center. And the results? They were night and day. The natural Colibactin, with its reactive, charged center, hit those AT-rich DNA targets with precision. But the stable neutral imposter? It ignored those targets completely. This was it, the conclusive proof. That unstable center was essential for aiming the weapon. So how does it work? It's actually a beautifully simple case of chemistry. See, those AT-rich regions of the DNA minor groove are naturally negatively charged, and Colibactin's unstable center, it picks up a proton, which gives it a positive charge. And just like magnets, opposites attract. This electrostatic pull is like a tractor beam, drawing Colibactin right in and positioning its two destructive warheads perfectly to handcuff the DNA. It's elegant, really. And with that, the case was closed. The specific positive charge drawing it to that AT-rich DNA, the precise spacing of its warheads creating that handcuff, everything about Colibactin's unique structure perfectly explained the criminal's fingerprint found in those cancer cells. The suspect and the signature were a perfect match. So, the case is cracked. But, so what? Why does solving this one molecular mystery matter so much? Well, because it helps us understand the hidden dangers that might be lurking within our own microbiome. So let's just trace that full chain of evidence one last time. It all starts with PKS E. coli in your gut producing this unstable Colibactin toxin. Then, that toxin uses its positive charge to home in on a very specific spot on your DNA. It forms that handcuff-like crosslink. And then when your own cells try to repair this mess, they can make a mistake, creating a permanent mutation. And that mutation is the fingerprint. This whole discovery gives us a direct atom-by-atom link from a common gut microbe to the kind of DNA damage that can lead to cancer. It's not just a statistical correlation anymore. We now understand the precise molecular mechanism. We've basically witnessed the crime as it happens. And that leaves us with a final, kind of provocative thought. If evolution can produce such an elegant and dangerous molecular machine in just one common bacterium, what other incredible chemical secrets, for good or for ill, are still hiding out there, undiscovered, within the vast and complex world of our own microbiome?